Well, hey everyone, it is amazing to see all of your faces, as it is with each town hall. We have a special forum today. It's Reviewing Science 101, and this is predicated because we have a new wrinkle to our, for anyone that's attending our conference this year. Our posters will be being reviewed, and our best poster selection will be designated by us, the families, and not by colleague researchers. So we're the one doing the decision making. So today's session is learning how to hold those researchers accountable, to dive through and learn how to dig through their posters and learn about everything. But I think that this session will have value for even if you're not attending our conference and are interested in the science on how to look at a paper, what questions should you be asking? And it seems really complicated at that, from 10,000 feet, but there's a lot of common themes throughout every poster and every publication that, understanding the anatomy and how we look and think through these questions, once you understand the pattern, it becomes easier and easier to go through these publications. So we will kind of go through these wrinkles today. Maybe. There we go. All right, so the first thing is the anatomy of a poster or a publication. So just about every single poster or publication falls under a very similar structuring. And the structuring elements or categories are the introduction, the objectives, the methods, the results, and the discussion. And regardless of whatever project they're testing or whatever questions they're trying to address, whenever they report their data, it's often this identical same format. And understanding this format and what each of these sections mean helps you kind of get through the meat and potatoes of what they're asking and what they're trying to tell you. So kind of going through each of these one by one, the very first is the introduction. So the introduction is really supposed to be high level of what they are studying. So for us, it's often an SCN2A or a tangential area of science or anatomy. But it's also why they are studying it. Just studying SCN2A is interesting, but they have an angle or a take that they're trying to study and understand. And the introduction will be how they're framing their question. But they're also discussing what they know, what the field knows, kind of the broader they, not just they themselves, but the broader field knows, and also what they don't know. But the combination of kind of these four, why, what, frame something that we often call the gap in knowledge. And that gap in knowledge is supposed to be field level. So what the field of science doesn't know about a particular question. And for SCN2A, we're slowly closing all those gaps to better understand the disease and the biology. And the hope is that better understanding will help us be able to design better drugs, therapies, and also clinical regimens to better be able to treat our children. So for this section, whenever you read a publication or a poster, you want to focus your questions around some of these things in the box here of really focusing in on that gap. What is the gap? Why is this a gap? Have them really kind of go through and explain it to you of why they think that is a gap and why it's important, and why should you care? Not just us as a community, but you as a parent. It's your job as the reviewer and critiquer of their poster, or any research. They have to explain it to you of why it's important for you and why you should care about it. But also, what would happen if we do know this gap? Like, how does it move the field? So this introductory section of kind of reading through that and kind of going through with the main focus on what is the gap in knowledge. That should be your takeaway when you leave the introduction. But a pro tip in the introductory section is researchers are like notoriously nervous presenting. I see Karen smiling because I think all of us researchers, particularly when you're a junior investigator, are incredibly nervous when you're up there in front of your poster. But the funny thing is we all get less nervous when we start talking about our work, because that's something we're familiar with. We spend all of our time in the lab and not communicating with people. So, talking about the science and what we're working on gets us back to our comfort zone. But they're there presenting because they care. They care about their research and they care about the impact that they're trying to make. And wanting to explain their work and find meaning in all the time and effort that they're spending in the lab, slaving away is really important to them. So, talking to you, a caregiver of a child that has a condition that they're studying, I think brings incredible value to, their efforts on a day-to-day. And ask questions, make them explain each and every little part. We always say that question, there are no ridiculous questions, and there aren't. Have them break down into small little chunks. And it helps them too, because every time they're asked to explain it in a different way or a different scope, they're then able to better understand what they're studying. And often even very simple questions asked a different way give them a different perspective on their work and their research that can help them learn, understand, and do better research. And also as they grow in their career, they're kind of, they'll have to, at first they're really deep in the weeds, but over time they'll be forced to kind of explain their research and science to different audiences. And these poster presentation opportunities are a great place for them to learn those skills. So get up there, ask questions, and don't be afraid to ask them to learn more about things. Objectives. So not every poster will have these, but many do. It's nice when they do have it and kind of lay out what the gap in knowledge is there for you, very easy to digest. But it's essentially kind of what the gap in knowledge is and what they hope to address in that gap. The methods. So the methods are, I'm a sucker for a method section. In my opinion, I think this is like the most important part of any research, any poster. It talks about the how, how they're addressing the question that they're trying to evaluate, and how they're going to answer the gap that was framed in the introduction with experimentation. And methods, are really what distinguish good science from bad science, and also great science from good science. So it goes into what model you are testing. Why is that the best model for this particular experiment and use case? Is that model well understood? These are questions that you can take verbatim and throw at the researchers and see how they'll respond. What assay or test they're using. An assay is just a fancy word for test, and it's how they're evaluating the experiment and the question. And why is that the best assay for this particular experiment? And the things that you should be looking for when they talk about that are, this is a really well understood experiment or assay. Everyone uses this, where it's very well understood in the field. And that gives you confidence that it's a good experiment to address the question that they're answering. If it's a very novel assay, there could be some risks around the technical implementations of that assay that make maybe some of their interpretations questionable. Also, with this assay most used for this particular experiment, also what time points are they looking at? I think that's a good question to always ask with any experiment, because with most questions that you're asking, things do change over time. And having, they may not have the best answer, but having them shown that they've thought through that question of, I'm looking at this time point because X, Y, and Z. Them having an answer like that, I think, always adds conviction to the results that they're trying to explain. But I think a really good study often asks the same question in a lot of different ways. So a good question is always asked, are there other ways to test this exact same question? And are there other models to validate these type of experiments? In a way that I can maybe explain that better. Excuse me. Is if you're trying to measure your table, there are probably a few different ways that you could measure your table. One could be, a measuring stick. It could be a ruler. That you have to kind of move throughout the table and count your number of rulers in inches that way. But in the end, you'd hope that you'd come to the same number of inches or centimeters at the end of your table. And if you don't, maybe that suggests that either the rulers or tape measure that you're using isn't the best way to evaluate the question that you're asking. So testing the same question with multiple assays or tests gives you higher conviction that the results that you're finding are actually true and accurate. And having experiments and posters and presentations that test the same question in multiple lights raises the rigor and quality of the research that you're evaluating. So
Sean, can I just interrupt you just for a second? I'm so sorry, but somebody had a question about how do you know if it's a novel or regular assay? How can they decide that?
So if you're in front of a poster, it's a great question to ask. These conversations with the presenters should be, informal. Ask them questions like that. How often has this been done? Or is it new? Is it novel? It's a great way for to engage in the conversation and evaluate their knowledge. If you're evaluating a paper, it's a little bit trickier. You can search the assay that they're talking about on Google and see if it's something that pops up, regularly. If it's something that's done a lot, there's probably like a Wikipedia page explaining how to do the assay and what it means. If it's something more novel, you may have to kind of dig to even find a description of that. And that would suggest that, novel isn't always bad, but it just means that you have to evaluate the experiments with higher degree of rigor in order to have conviction that it's good.
Could I just add to that, Sean?
Yes, of course.
So sometimes there's nothing wrong with a novel or new assay, and sometimes it can really make the field leap forward. It could be very exciting. But when they say this is a novel assay and we've developed in our lab, one way to evaluate how trustworthy it is, how rigorous it is if they say, and then we tested this novel assay against a well-known reproducible assay that everyone uses. So if they've benchmarked it against something that they know works and gives certain answers, and they can compare their answers against a well-known assay, then that also gives me more trust in the new assay.
Absolutely. And maybe a way to kind of think about it in a real life scenario is we recently got some blinds for our house. And we got blinds for our whole house. So we just got a new house. And I at first was going to measure all of them with like a tape measure, and like, God, this is going to take absolutely forever to do. And I wasn't sure if I should be going to like the 30-second or what, and had a piece of paper and I was writing it all down. Like, this is just crazy. So I ended up going to Home Depot and getting a laser laser measuring, where you just point the laser and it tells you the distance from X to Z where the laser points. I'm like, oh, this is so cool. Like, this is going to cut down the time so much. But then I'm like, oh, is this even accurate? I'm not certain. But the experiment that I did to confirm its accuracy was to use the laser level and then get out my tape measure, kind of old trusty dusty tape measure, and then measure the same area. And to my happiness, that they came out to be the exact same amount. But if they didn't, if they were different, that would suggest that there is some flaw in the new assay. But we have to do the exact same thing when looking at different aspects of cells or animals or behaviors. But I totally agree with Karen. And even with this example, like, this was a novel way to measure a laser that cut down on time, and probably was more accurate than the tape measure over a lot of different measurements. But you still have to do that level of validation that it's correct. But what are models? So there are a lot of different types of models, and you don't have to know all of them, but you should understand kind of roughly what they are and what they mean. So models are a system that we're asking our question in. And for biologists or most of our researchers, that this usually in a biological system. So it could be a cell. Often for us, it's neurons, or cells that are, have SCN2A in them in a certain light. But questions that are good for you as the reviewer to understand are, what is the model? Is it a cell? Is it an animal? If it's a cell, are they growing these in a dish, or are they growing these cells in an animal? Also ask, like, where they came from. Are these cells from an animal, or are they from a patient? And depending on their source of origin and how they're growing it, it changes how you evaluate what that experimental system is and how they're asking the question. And don't be afraid to ask, like, okay, this is a cell in a dish. Like, what does this mean for the experiment you're asking? Again, is this the right model for the question that you're trying to ask? So it always comes back to when you're the reviewer of a poster, of asking more questions. It never hurts to continue to drill down and understand more about what they're presenting on. For animals, it becomes a little bit more interesting. So there are some prototypical like animal models that are used in SCN2A research. And it's often mouse, rat, There are some that do voles. I'm happy to talk about voles if anyone is interested. And then there are, interesting that we are able to actually engineer these mice and rats to be different than kind of the mouse and rat that you see out in the field, hopefully not in your house, but unless it's a pet rat, that's a different story. But if you catch a mouse outside versus the ones that are being done in the lab, the ones in the lab are often engineered to do something in a particular way or in a particular fashion. So they may light up if a certain laser is shined on them. And the way that they're engineered allows us to, ask a question in a very specific way to get a particular answer. And for us in 2A, it's often about expressing or changing the expression of SCN2A to either a particular time period or a particular tissue. We know that SCN2A impacts our kiddos in a lot of different ways. And having it throughout, like expressed throughout the whole body, makes it hard to ask certain questions because the whole body could be changing the way that we see it in our children. We change one little thing with our child and 30 other things change along the way. It makes it hard to kind of do experiments to figure out, for sure whether something is changed to that particular scenario. So we often engineer these animals so SCN2A is only expressed in a particular location at a particular time, so that we know if a change leads to a particular change in clinical phenotype, that it's due to SCN2A change at that particular time and location. It gives us higher conviction and rigor to our experiment and our determinations at the end of the experiment. We also do experiments in humans. Sometimes those are without a experiment. It's just kind of a learning experiment, how our children are reacting, how they're growing, what they look like. Those are still really meaningful experiments and models that people may be presenting on at our conference or in a publication. And those describe what our children look like on the day-to-day, how they're responding to medications, what maybe they have osteoporosis or something else that we should be on the lookout as parents. Excuse me. And asking our doctor, like, is this something that I should be, thinking about for my child? And there certainly will be posters on humans, and natural history is often called within our community. Or when we get to therapeutics, they're often testing that new drug in some patients and then not in others, and evaluating whether that drug is both safe and effective. And there's also in silico, which is a fancy term for in a computer or in a model or an algorithm. So we're getting to a point now with computers and AIs where we can, ask some questions and get a fairly reliable answer that it would be similar to doing it in an experiment or a dish, much faster in a computer. And being able to do those quickly and rapidly allows us to generate new ideas and hypotheses that we can go back and then test in the lab. So going back to our anatomy, the next section is our results, and these are the findings from the experiment. So before we talked about the methods, which was how we're testing, the results are, that they actually took that how and did those, experiments in those models with those assays, and these are what they found. Often results are presented in either figures, graphs, images, or dioramas. And it's their way, and the results can often get very technical, and don't get intimidated by that. It's totally okay to ask them to distill, this result, translate this to me, explain it in a way that I can understand. Talk to me like you'd explain this to somebody that doesn't know deep science. Challenge them to explain their research and their experiment in different ways. But we always say the results are the results are the results. So the results section is really supposed to be not interpretations, but a presentation of their findings. And the presentation of their findings may be positive, they may be negative, but they're supposed to be, we ran our methods, and these are what we found from these experiments. So while you're evaluating the results, it's important to keep in mind that gap of knowledge that we framed in the early introduction. Look at the experiment and the results and see how it fits within the broader story. So in the beginning, we were kind of framing, what are we studying, why we're studying it, what is the gap? When you're evaluating these individual nuggets of results and asking the research to explain it, take their explanation and then kind of frame it in your head in the broader story that we were asking all along. But also look for oddities in each of the figure. Look for things that don't look right and ask them to explain it. So if you see a cluster of dots in one section and then a few way out on the side, ask them where those dots are on the side and why those are out there. Do they have a good explanation for it? They may not, but understanding and kind of diving into the results section allows you to better understand the story and how reproducible it is. And also allows you to evaluate the researcher and how much thought they put into the project and their level of conviction. And also be a cool kid. Ask, is it stat sig? And stat sig means statistically significant. And that's a fancy way of meaning that they've done the experiment enough times to know that it's unlikely to be a byproduct of randomness, the effects that they're seeing. But stat sig's a slang way of asking that. An event move, you could also, this is one thing I always do with posters. I always kind of do active listening. And I always ask, so you're saying, and I try to repeat back in a different way what they've been trying to explain to me. And then I always follow up with like, okay, so pulling it all together, ask them to kind of keep repeating and keep explaining their results in different ways and different manners. And test them to see how consistent they are with their explanation. And if you notice any oddities in their explanation, then call them out on it. Last time you said this, but this time you're saying this, like explain that to me a little bit more. And each time they explain it, you get a little bit better picture of the story that they're trying to tell. The discussion section is an important one. So the discussion section is supposed to be the researcher's interpretation of the results. So it's in their opinion what these results mean. Did they close a gap? Did they close that gap in knowledge? Were their experiments successful? Did they validate or nullify their hypothesis? But also why these are important. Were there technical limitations or implications of the experiments that they were doing? And it's also their opportunity to talk about how their research fits into the bigger picture, not just that kind of the small gap that they framed, but the bigger picture of SCN2A or maybe autism or epilepsy. If it's a big paper and big findings, often those findings that they determine can have really big implications on how we view SCN2A, epilepsy, so some of these big terms that we all know about. But they should also be talking about what they plan to do from here. So our experiments led us to this point, and this is the next logical place that we should be testing or evaluating. And sometimes it could be, our results in this poster didn't really tell us what we hoped. And we have to kind of go back to the drawing board and think about it in a different way. And that's totally fine. In full transparency, often that's what science is, keep reevaluating different ways to test these questions that we're trying to find in a way that we can finally get reproducible and reliable results. And that reproducible and reliability is really, really important for science. It's when you're able to get that high degree of rigor, reproducibility across multiple models where you can finally say, this is true, this is a true phenomenon that we can then interpret and understand deeper into what we're evaluating, which is, the biology of SCN2A. But this is also, the discussion is your chance to really infuse the voice of the patient into the chat. So they're going to be thinking about things very much in the scope of an academic researcher of like, that these results mean this about this technical nuance of the biology of the protein. You're like, I don't care or know anything about that. What I care about is what does this mean for our children? So like change the discussion and put it in the tone to what it means for you. How did this translate? We often say, how do these academic findings translate to a better treatment or a better cure or a better clinical approach? Keep changing the conversation to turn it into the voice of the patient. And ask them, can these results be used for a treatment or improve our care? And these conversations that we're having with particularly junior investigators are really important to kind of help frame how they think about their science and how they think about their results. Because the next time they go to the drawing board and think about their next experiment, this conversation that they had with you, and then they maybe meet your child, instead of maybe doing a really deep dive into a more academic question, maybe it shifts slightly to a more translatable question that could lead to a treatment or a different way to think about things clinically. So these interactions that you're having as a caregiver with these researchers are really important for kind of shaping their mind and future decision making in our space. we play a really important role in researchers' next steps. So some other things that I think are important to think about and to try to learn during these interactions that I think maybe we don't think about a lot as caregivers. Ask them how much all this cost. Science is incredibly expensive. And I see Angie on there. Angie often likes to talk about hope has a cost. And I think that's such a brilliant line. But try to frame it, like they did these experiments and I took time to understand them. And now I understand what they mean. But how much did all this cost? And how long did it take? And try to get a better perspective of, how much these like often can be little but important findings that are kind of building Legos into a bigger picture of SCN2A, how much each of these little Legos kind of cost and how long they take to kind of understand the Lego and have high conviction in it. And then put that to the bigger picture of what we're trying to build, which is like a complete picture of SCN2A and also how we can, infuse tools into that Lego diorama that we're building of SCN2A in order to change how it impacts our kids clinically. And an important thing to know is like we don't need to have the full picture in order to develop treatments and cures for our kids. But each new Lego that we have better informs how much we're able to design and develop new drugs. But also understand like a little bit more about them as the researcher and the person. Understand like how much time they spend in the lab each day. I could tell you, firsthand when myself and my wife were getting our PhD, like we literally never left the lab. It felt like we slept in there every day in order to like take out when we could. But understand the perspective and how much effort and love they put into these experiments that they're doing to try to save our kids' lives. And also ask them what the typical day looks like. Are they on the computer? Are they handling 300 mice and they're getting bit by their hands because the mice are angry with them, or what they got going on. Help understand what they're doing on a day-to-day. But also understand kind of the lab dynamics. Who is in their lab, who else they're working with. Are they in charge of their lab? Understand kind of what goes on there. I think the most important question to ask is if they've met any 2A kiddos yet. And have they met your kiddo? And if not, definitely introduce them. Our kids are awesome, and it's important that our researchers, have met them and learn how incredible they are. Other things that are important to know about are like what they're working on in their lab next. Do they have something cool and exciting that they're working on? Is it funded yet? And if they're a young investigator, what do they want to do with their career? Often when you're a young investigator, you think about those things, but not totally. And sometimes these single interactions at conferences can have, huge implications on how their careers develop. And every new researcher that we have studying SCN2A helps build us and collect more Legos to put together the picture quicker. So, us as a community want to recruit as much talent as we can into our research ecosystem as fast as we can. So these interactions with you as caregivers can be really meaningful to help us, build the best ecosystem that we can. I will pause there. A lot thrown at you, and also go back to the poster if anyone wants to ask any questions on anything, or if anyone wants to jump into any element. We could definitely get more technical too on different assays or different models, or we could jump into anything y'all want, but I wanted to keep it a little bit high level at the get-go. And I see Karen raised her hand.
So I love this presentation, Sean. It was so beautifully done. I wish that when I was a first year graduate student, someone had given this presentation to me, because it would have made all of my poster attendances so much more productive. So thank you so much for that.
Yeah,
I
sure.
did just add that sometimes the label of the sections will be slightly different, and that's okay. It's always going to still be presented in this order, and the sections will still be have the same material that Sean laid out in it. For example, an introduction could also be labeled as an abstract. The abstract is a tight summary of everything that they're going to go over in the poster, as well as the kind of introductory, this is where this question lies in the field. So you might see the word abstract. It's the same place to start. It's like the introduction. The objectives might be called aims. Objectives and aims are used interchangeably in science. And so if you have a goal, for the work, then it could be called an objective, or it could be called an aim. Materials and methods are usually always called materials and methods or methods. And then the discussion can also be the interpretation, or it could also be labeled conclusions. And that conclusion section still means this is where I'm going to discuss what the results of this work means in the bigger picture sort of way, the exact way that Sean explained it. So don't be dismayed if you see completely different words on a poster, which I know some of the posters I've made will have different words on it than this, but they're exactly the same and they mean the same thing.
Thank you, Karen. The first poster I pulled up had different sections on it, and it was mine. And then I felt bad that they were confusing, so I didn't use it. So I used aims, which is more classic. I see Christina has a question too.
It's more of a comment, but one of the things that you brought up in the very beginning was how some of the young investigators or early career researchers get really nervous. And I think that that's a really good opportunity for families to find common ground with the researchers and just sort of let them, obviously framing it as like, this is why we're here. Have you met a kid with SCN2A? Here's my child, which is a great suggestion. But also maybe ask them why they were interested in SCN2A, what got them started. Sometimes, it was the work of their mentor that got them down the path of SCN2A, and then maybe they fell in love with the science. Or maybe you'll hear a story with a researcher that said is actually, I attended a conference for families SCN2A last year, and I fell in love with the families, and that's really what has lit a fire under me and kept me going. And I think finding that common ground will really get the researchers excited, and maybe make them a little bit less nervous. So that was one comment. And then the other, which again, Sean, you did such a great job sort of explaining everything, but I love that you used analogies, especially like the curtain shopping, which I love. But if you ever I always love analogies because it really does distill down sometimes really complicated things in easy to understand terms. So don't be afraid to ask them, hey, can you put this in an analogy for me? And we'll try to prep the researchers too to have an analogy ready for sort of the really complicated stuff, because it really does break it down in an easy to understand way. So have that in like your back pocket, ready to ask them for that analogy if it's over your head or really difficult to understand.
Dan, the man. Hey, bud.
My questions aren't overly complex for you, buddy. I actually prefer if they talk to me like I'm in second grade. That's what I ask everybody to do in my life. Just pretend I'm a second grader and tell me like I'm in second grade. But my curiosity, I'm trying to sort of understand like the process. So a researcher who works for a lab is doing the work. If the experiment, let's say, is successful, then the goal, I assume, from there is that some pharmaceutical company or someone from there then takes sort of the next step to try to put something together to help our kids. Am I kind of understanding the general big picture process behind all this?
Ish. I will say ish.
Yeah.
And the main reason I mean ish is that researchers can kind of fall into a lot of different buckets for most that are presenting posters. Like some are purely interested in academic questions of like, how did this protein work or act in this scenario? So like those are important into understanding, but have a few steps before they convert into a therapeutic. There are also researchers that are highly translational. So their main focus of their lab is to evaluate therapeutics in the context of the biology. So our translational scientists are the ones that really are evaluating a lot of different drugs or ideas or molecules, looking for rigorous that same concept of reproducible effects that change the biology back to normal as much as possible. And when they identify something that reproducibly does that, those ideas then often mature up into a biotech company that can take them and spin it off into a company that's sole goal is to develop this into a drug for our kids. And biotech companies themselves often are highly research based as well. Often doing the same thing as our translational scientists, but at at a bigger scale with a lot more money. So I think, conceptually, it's probably think about them in the three different buckets of, more academic based, translational, and then drug developers. And I think one area we have an opportunity in forums like this is to make our academic researchers, which are really going to be the base of your ecosystem, where you have maybe the most number of researchers, like give them that translational mindset. So when they have an opportunity to ask maybe a more academic question, they ask it in a more translational way. So to your point, when they do have a finding, those findings are more likely to translate to therapeutics or things we could translate to benefit for a kid.
Yeah, good. Thank you for that answer. I thought, there's an end goal here. I'm just trying to understand from start to finish, but it varies depending on who's doing the research. So thank you.
Yeah, of course. Jessica?
Yeah. So let's say we see a poster and it's correlated to our child's mutation or something that they have that we're like, yes, we could be a part of that. Oh, my computer's about to die.
Oh no.
The big question is, what are the next thing? Besides tell me more, how do we help? How do we get our kids involved in this a little bit more? And is there a best practice protocol for that, as opposed to just like giving them all of our kids' DNA and then begging for results in a couple of months?
Amazing question, Jessica. Yes,
it's like I've accidentally done it before.
Yeah, I think that is something that probably a lot of us have maybe encountered. And the best practices question, I think, is really important. I think our job as a patient advocacy group is to play that middleman, between our researchers and our community, in order to make sure that things are being done, in the best and most ethical and most appropriate way for our community at large. So maybe when in doubt in those scenarios, don't hesitate to reach out to Karen or Christina or myself, and we'll definitely help you kind of guide in those instances. But definitely can talk about your child. Like, I think a big benefit for researchers that are coming to these engagements are to learn about 2A, learn about children, learn about their phenotype, sharing descriptives of your child. I don't think there's, any bad way of going about doing that. But if you start sharing like medical records or things that have HIPAA elements or biological data, it's like there you want to make sure that there are, different protocols that have been approved, that show that the research is being done ethically and appropriately. And we can help you navigate those waters anytime those are being found. But also, if you see something cool that's interesting, I'll also raise it to us. Like, I think our job is also to be, to help kind of keep pushing the science and research along. If something seems interesting to you and you're like, man, I think if they had my sample, this could really move things along. Elevate those things to us, and we can keep pushing the researchers too, to make sure that they have their IRB in place, that they have their protocols in place to do those in a quick, rigorous, and appropriate way. Karen, any thoughts that you think are important to mention in that scope as well? I know you talked about this idea or concept at a recent town hall. Oop, unmute.
Sorry about that. Yeah, that's a super interesting and very important question. There are ethical guidelines that, in fact, our bioethicist Melody should be actually answering this.
Oh, there she is.
that need to be followed. And I think Sean is right on the money. We're all here to help. And Melody is an expert in this. I'm going to back off and let her spit.
No. Well, no, I don't want to take from you, Karen. I think you've got tremendous amount of experience with this. But what part can I help with? What would be the most useful thing that I could share from my bioethics background?
Well, I think the importance of checking to make sure that there's an IRB in place.
Yeah.
And make sure that there's a design in place where, for example, a lot of genetic testing companies will take your child's samples, go and analyze it, publish a study, de-identify everything, and never be able to give life-saving data back to a child's family, saying, by the way, did you know that you had a gene change that could lead to blindness later on in life? I mean, these are the kinds of things you need to understand. Are the results going to be returned to you or not? Is this data going to be belonging to you as the participant, or will the data then be siloed off in some academic or other kind of center and never to be released again, except maybe for millions of dollars to a pharmaceutical company? You know, these are the kinds of questions that I think need to be asked before at least so that you know before participating what the benefits are, right?
Absolutely. And I would say also the opposite of what you said. It's really important to know what won't be shared. And sometimes you would want your child's information to be shared widely, but because of the nature of the consent form that you signed when you joined a patient registry or donated a sample of cells or however you participated, the consent form may not allow that research team to then share your data with another group that could advance the science further. And so you may be contacted to be either reconsented to share with a different group, or possibly not contacted at all, and then it doesn't go forward. So I think those are great questions to ask. And also, that's actually one of the things that we've been working on for there's an informed consent section of the new website when it comes up, and asking questions like, if I participate in this research, will that preclude me from participating in future research? Or what implications does that have? What questions should I ask for participation? Do I have choices? Can I wait? There's all different kinds of questions to ask. And so I think it's a really important aspect of participating in research. And when you're looking at a poster, asking how was this research vetted? Who looked at it, and with what lens? Really important.
Maybe to recap, or pulling it all together, these things can be very complicated. And a big part of our job and role and responsibility is to evaluate those, and to make sure that the data is being publicly shared as much as possible. That
Okay, baby, go potty.
Are being shared. So leverage us whenever you have questions. We're happy, and we're there for you all. Yeah, particularly when we have people like Melody and Karen that, so smart and sophisticated on these elements.
And you, Sean.
I think that's actually one of the great uses of advocacy groups, is to be that bridge and to have a second set of eyes. You've got the ethics review board, or the IRB, looking at this, approving the science and the research, but you also have the advocacy groups asking these tough questions, or especially when it involves like a clinical trial or an investigative intervention. You have the advocacy groups advocating for families to make sure that they're not promoting a study that wouldn't be in the best interest of the families. So it's a great use of advocacy smarts.
Melody, you have a really good point about results not being returned. So that happens a lot, where we participate in research thinking that we're going to get something back from it. And most of the time, you don't get anything back from research. You're contributing to a greater body of knowledge that's going to advance the field. And we should walk away and feel good about that. But I think it's really important to set those expectations up front. And if it's not made uber clear by the scientist or the coordinator, we should be asking those questions. And thank you for empowering us to remember to ask those questions, because sometimes we feel intimidated, like maybe we won't be eligible to participate if we ask. We're just grateful for any opportunity, and we just stay quiet and just sign away. But it's important to know.
100%.
So I have a question. So Sean, you talk about the scientist being shy, a young investigator being shy about explaining their results. But sometimes the families are the shy ones, right? So like, get any advice for some of us that, and obviously it's not me, but that has a hard time asking questions or feels shy about going up to a researcher. Yeah, go ahead, Karen.
There was one point that I really wanted to make about this, and that is that, yes, the presenters are going, especially the young ones, will be nervous. But they're also, I think, they're hopeful that someone will come by and want to listen to their work because they have devoted years of their life and weekends and days in and days out and skipped meals to produce this work to show you. And so I think there's nothing sadder than someone standing by a poster and everybody's walking by them and no one's talking to them. And I mean, that's been me. And I feel like I'm less nervous. I'm more excited to share. And so keep in mind that there might be some nerves involved, but if you really are interested and you just want to go and listen to their presentation, that is a thrill for any scientist. It really is. And so you're doing them a great favor. And hopefully that helps with the shyness. If you are really interested, but also feeling like you want to grab one of us, then grab one of us and we will happily go with you and be by your side and ask the questions for you.
That's a great idea.
Another vet move is to kind of like sidle another conversation that's going on and like listen to the question that they're asking and then just kind of jump in mid conversation if something is interesting, or you can go back and ask the exact same questions and to break the ice. I think that I've definitely done that technique before.
Can I just go back to the very important point that Dan made with his question? I wanted to say that basic science feeds translational science. So I say this as a translational researcher at a drug company myself. Sometimes there's so much of a gap in the basic knowledge of how the disease works, how it looks like in a mouse, what happens to the cells when you've knocked out the function of SCN2A. There are sometimes these basic questions that are not answered. And so even if it's a basic question that gets us a little further along that basic question path, it could be essentially a very important answer that is then taken up and used by a translational scientist later. One of the big dearths that I find sometimes in the literature is questions that I have about the basic science. How can I tell if this drug that I'm developing is going to work or not if I don't even know what it is that I want to correct about the disease? And so basic science has a very fundamental place in the whole network, in the whole ecosystem of finally getting to a treatment for our children. And so if you happen to stand in front of a poster and find out that it's basic science, I would say, please don't dismiss it because it could be extremely valuable later on. It's just like, it's not going to directly lead, by its definition of basic science, it's not going to directly lead to, well, here's a drug that we're going to be able to put into people tomorrow. It's just, that's not the nature of how science is done. But fantastic question. It's good to have that framework in your mind.
Thanks, Karen. We definitely see that a lot. You know, talking with the drug developer, they often come to us saying like, God, I wish I knew this. You know, does anybody know this? And, you know, we're often connecting them with an academic researcher that knows this or knows how to do that and answer that question quickly. But those are, you know, essential cards that have to be flipped in order to, you know, make a thoughtfully designed therapeutic. Maybe one example is like, we still don't know how long. It's called like half-life, but once SCN2A protein is made, how long it lives in the body before a new SCN2A protein has to be made. Like we don't know that answer. And for people that are trying to design, you know, drugs that can make more, it's a really important question in order to be able to math that out in the right way to have just the right amount of 2A that it could be beneficial. But in its own right, it's not therapeutic driving. But it answers fundamental questions that are imperative for drug development. One other area that we didn't talk about too much are like clinical research and natural history. We did talk about it a little bit. But to Dan's question, we think about, academic and translational science, but we also have like clinical scientists too. And their job is often to figure out how best to treat our kids with what we have at hand, and what the disease looks like and smells like at every age that our kids could be. And those could be really interesting and meaningful research for us as parents, because those can often have actionable items for us today. We were talking at an earlier meeting with the team today of, after the one conference I left in Harper, I went back and asked questions to our neurologist, and Harper ended up with, 2 new diagnoses that she didn't have before. And it was all because I learned about, these 2 other things that some SCN2A kiddos had, and I didn't know about them. And then when I pressure tested our neurologist, she's like, yeah, they may, let's go get tested. And sure enough, tack it on to our long list of diagnoses that Harper has. But I think some of those research can be really interesting for us as caregivers as well. Any other questions? Has anyone read a SCN2A science paper before?
How about we say attempted to read? Is that a better analogy? I was actually trying to put it through AI and make it write it in like easier language. It kind of works. Like, it's not perfect, but you can kind of see like, okay, it helped understand a little bit.
It's a great tool to get through things quickly. And ask it to Christina's point, give me an analogy of what this means, to translate it in a different way. And it's a great way to kind of digest things really in different thoughtful ways. Would it be helpful, Dan, if we, at a setting or town hall like this, to go through an actual publication and understand where the pain points are for you guys to digest those things? Is that something that they'd be interested in?
I mean, I would love it. I could listen to you guys talk about this stuff all day. Like, I think it's just great. Like, I'm just listening. Like, this is so cool. Like, I'm understanding how this world works. That this big, broad world. But 100%. I mean, that would definitely be helpful.
Cool. We'll tee that up for a future town hall then. Maybe we'll put a survey out of what type of journal would be fun for y'all, and then we'll pick one and then we'll rip it apart.
Awesome.
I love that. Dan, I'm actually working on some lay summaries of some of the key SCN2A articles right now, and I'm going to learn from tonight and put some analogies in there. I think it's a great way to learn, and I think it might really help folks really understand the material. So if you have any other ideas for what's the best way for you to learn, or what would be the most useful summary information, please share with me.
Oh,
Melody,
Mel,
just remember, I'm very low functioning, and I love I think Karen said the abstract. I'm a big abstract guy. You give me an abstract, I'm in, man. That's my zone right there.
I love it. I think one of the hardest things for me is I'm shooting to hit it for 3rd grade level, which is actually incredibly hard. There are some words that just literally do not translate to 3rd grade. But we're trying. I'm shooting for 3rd grade. I'm probably going to hit 7th grade, but I'm shooting for no scientific knowledge, no heavy vocabulary.
Yes.
I think I hear some parents, perhaps Dan, on this call, volunteering to be a committee, like a parent committee on evaluating some of your lay summaries.
Oh, Karen, that would be so wonderful.
Melody, I do whatever I'm told. You can ask my wife, she's over there. You just tell me what to do. You send it to me and I produce. You just say, Dan, this needs to get done by this date. I'll probably wait till like 4 minutes before it's due, but I'll produce something beautiful in those 4 minutes. You'll see.
Excellent. And I'll probably send it to you at 3 a.m, so it's all good.
We're a great team then. That's how I like to operate. So that's great.
I love it.
I love you, Dan. Thank you.
Dig it. Dig it. Who's going to the conference this year? I see some hand raised. Who wants to evaluate some posters at the conference? All right. This is recording, Leah. We gotta keep tallies here. Cool.
Amazing.
Dig it. And if anyone has any other questions or wants to dig into any other elements, we're here and happy to talk through anything.
Sounds good. Thank you so much, Sean. Thanks everyone for joining. Have a great night.
Thanks for all your hard work, guys. Have a good night. See y'all.
Good night, everyone.